of you. Today we will discuss about the transcription. So before going into that, just we have to look on what is the central dogma. Okay, so central dogma is a flowchart of how the DNA is converting into the protein. Okay, so this is firstly we are having the nucleotide sequence, which is called as a DNA. Okay, it is the information storage, and from here by the process of transcription, the DNA getting converted into the mRNA. Okay, it is the just replica of the DNA, but it is more like in sense form, right? Like it is having the coding as well as like in DNA there is two parts. One is the exon, another one is the intron, but in mRNA there is only coding part will be there, which will code to the protein. Okay, the exon part, intron will be removed while the processing and all. Okay, so this DNA when codes for RNA, this procedure is called as the transcription. Okay, and this then this RNA when converted into the protein, then this process is called as the translation. And this complete flow chart from DNA to protein, this complete flow chart is called as the central dogma. Okay, so here we can see that this is the two strand of the DNA. One is the 5 dash to 3 dash end, and another one is the 3 dash to 5 dash end. Okay, and this is the mRNA sequence. So this mRNA sequence is identical to, always identical to the coding strand. Okay, and it is complementary to the template strand. Okay, so just remember this thing, like mRNA, the whatever mRNA sequence you will get, that will be the exactly identical to the coding sequence of the DNA. Okay, it will also start from the like 5 days to 3 days in. Okay, and it will be complementary to the template strand, right? And the only difference between this identical sequence is, in case of DNA there is thymine, and in case of the mRNA there is uracil. Okay, rest of the nucleotide will be same. Okay, in case of DNA only it is thymine, and in case of RNA it is uracil. That is the difference, and it is very much means it is completely identical with the coding sequence. And this coding strand is also known as the sense strand. Okay, and this template strand is known as the antisense strand. Okay, so this is the conversion of DNA to RNA. Okay, and it is, this procedure is called as the transcription. So there is a different type of the RNA and their function. So the most common type of the RNA is mRNA, rRNA and tRNA. Okay, this mRNA is called as the messenger RNA. This rRNA is called the ribosomal RNA, and this tRNA is called as the transfer RNA. Okay, and this mRNA is useful for the protein synthesis. Okay, because the protein, the amino acid sequence is forming from the mRNA itself. Okay, and this is this rRNA is ribosomal RNA, but it is code for only those protein which is produced from the ribosome. Okay, so this is the ribosomal rRNA, and this is the tRNA, which is helping in the charging of the amino acid during the protein synthesis. Okay, and apart from that there is regulatory RNAs is also there, like siRNA and miRNAs. Okay, it regulate or it silence the overexpression of any kind of transcript. Okay, or translation procedure. It will regulate through the siRNA, small interference RNA, or the miRNA, through the miRNA. Okay, and apart from that there is the small nuclear as well as the small nucleolar mRNA, which is helpful in the mRNA processing or rRNA processing. Okay, this we will see in the after translation, means once the transcript is produced, that time the processing should be happen for removing the intronic part. So that time this RNA play role. Okay, and in inside our body, when we wanted to silence particular protein expression, that time we can use this siRNA and miRNA. Okay, So, what is the transcription? So, transcription is the synthesis of one RNA molecule using one of the two DNA strand as a template by the enzyme RNA polymerase. Okay. So, this transcription happen from only one strand of the DNA, not from the two strand. Okay. And which strand will act as a template strand, which will be like 3 days to 5 days end. Okay. And the 5 days to 3 days end will be identical to the RNA sequence. Okay. So through the enzyme RNA polymerase. Okay. So here you can see there is two strand. Okay. And this orange color round shape is called as the RNA polymerase, and this is the start of the transcription, and this is the direction of the transcription. Like in case of DNA, the direction of the replication was 5 days to 3 days in. So here also the direction of the RNA synthesis is also 5 days to 3 days in. Okay, so the lower strand is called as the template strand, and the upper strand, which is from 5 days to 3 days in, it is called the non-template strand or coding strand or sense strand. Okay. And here we can see there the light green color is the newly synthesized RNA, and in this form this is called as the RNA DNA hybrid. Okay. So in like in case of the prokaryotic transcription, it is very much different from the eukaryotic transcription, right? So because the prokaryotic is very easy system, so that's why here the only single type of RNA polymerase participate for the transcription of different type of RNAs. Either it will be like mRNA, rRNA, tRNA. Everything will be produced by the single kind of RNA polymerase. But in case of the eukaryote, there is different kind of like three different kind of the RNA polymerase there, which produces different RNAs. Okay. So in case of this is the in case of eukaryotic. In prokaryotic there is no intronic part, so that's why the mRNA is directly getting converted into the protein. But in case of the eukaryotic, there is so many intronic region is there. So after processing and splicing, there will be only exonic part, which is called as the mRNA, and it will transport from the nuclear, and then in the cytosol it will secrete the protein. Okay. So as I told you, in case of bacterial transcription, in this class we will basically talk about the bacterial transcription. Okay, we will not go for the eukaryotic transcription because that is very complex process, and in this class it is not possible to cover the those two means transcription together. So that's why here I will discuss more about the bacterial transcription. Okay. So the enzyme which is responsible for the transcription is the RNA polymerase, which is a kind of DNA dependent RNA polymerase. Okay, because here what we have seen is the RNA is producing from the DNA as a template, right? So that's why this RNA polymerase is DNA dependent RNA polymerase, and it don't require any kind of primer for the polymerization. As you remember, like in case of DNA replication, that time the RNA polymerase required the primase require the RNA primer, short RNA primer for the DNA initiation, right? But here the RNA polymerase itself is sufficient to initiate the transcription process. So it doesn't require any kind of the primer for the polymerization. Okay. And it required DNA for the activity and is most active with a double stranded DNA as a template. Okay. Here also there is no need to like firstly the DNA will got separated and then the transcription will start. It is not like that. Okay. So here the RNA polymerase will only unwind the duplex of the DNA, and meanwhile it will synthesize the mRNA. Okay. And the synthesis will take part from 5 days to 3 days in, and it is a magnesium dependent enzyme, so it requires the magnesium ion as a cofactor. Okay, and here it like in case of DNA polymerase, it had the exonuclease activity from 3 days to 5 days in, but in case of RNA polymerase there is no exonuclease activity. Okay, so because what I can say is the error rate is already reduced during the replication time, right? So this time the error chances is very low. Okay, so that's why here the RNA polymerase is not having this exonuclease activity. If it is occur, so it will persist throughout the protein expression. Okay, so there is no way to rectify those errors. So that's why for before going into RNA production, the cell machinery in the beginning itself, it will rectify all the errors during the replication. Okay, then only it proceed for the RNA synthesis. Okay, so yeah, so it is a multi subunit enzyme. This bacterial RNA polymerase is multi subunit enzyme. Okay, so here we can see that there is 6 different subunits of RNA polymerase. One is the alpha, second one is the beta, beta dash, then omega, and then sigma factor. So without sigma factor, this whole subunit is called as the core RNA polymerase. Okay, and once this sigma factor bind to this complete core enzyme, it is called as the holo enzyme. Okay, so this complete system without sigma factor, it can produce the RNA by using the DNA template, but it can't produce on a proper site. Okay, so for synthesis of RNA on a proper site, it requires the sigma factor, because here the sigma factor is working as a recognizer. It will recognize the promoter sequence. Okay, in the promoter sequence there is two type of thing. One is the -10 and -35 sequence. Okay, so it will this sigma factor will go and firstly search this sequence in the DNA sequence. Okay, and once the sigma factor find this sequence, the RNA polymerase will go and sit this particular sequence and start the initiation of RNA. Okay, so without this sigma factor, this subunit can synthesize the RNA, but it won't be any proper place. Okay, so for proper place to be synthesized the RNA, it required the sigma factor. Okay, and once this sigma factor added into this complex, this is called as the holo enzyme. Okay, and these are the function, like this alpha subunit will determine what DNA should be go for the transcription. Okay, and it will organize all other subunit of the RNA polymerase. Okay, and this beta subunit, after recognizing by the sigma factor the promoter sequence, this beta subunit will catalyze the polymerization. Okay, and this beta dash will bind and open the DNA template. Okay, and this sigma factor will recognize the promoter for the synthesis of initiation, and this omega omega subunit will help in the subunit packaging with the alpha subunit. Okay, so these are the function. In exams it can be also asked, so you have to remember like all the function of different subunit. So the main function of all these subunit is, one is the sigma factor, which is helpful in the recognizing the promoter sequence and start initiation of the RNA synthesis, and another one is the beta subunit, which is helpful for the polymerization. It catalyzes the polymerization. Okay, so these two factors are very much essential in the in case of RNA polymerase. Okay, so yeah, so in like in transcription also there is two phases, like in case of DNA replication. Okay, first one is the initiation, second one is the elongation, and third one is the termination. Okay, So this RNA polymerase, firstly, as I told you, this RNA polymerase go and search for the promoter sequence. Okay, the sigma sub unit will search for the promoter sequence, and what kind of promoter sequence it will search, which is having the TTGACA region. Okay, this is the consensus sequence. Okay, which is present in the downstream of minus 35 of promoter sequence. Okay, and as well as it will also find the TATAT region. Okay, which is also called as the 10 minus 10 sequence. Okay, and after binding here, here you can see that this is the DNA polymerase. Okay, so this is, suppose this is the promoter sequence. Okay, which sigma subunit identified, and after binding this here, what RNA polymerase will do is it will little bit open up this DNA. Okay, because it got the signal like from here we have to start the RNA synthesis. Okay, because this is the transcription start site. So after searching this sequence, it will start the synthesis from here. Okay, and the RNA will quickly what I can say is terminated. Okay, so means there is no primer before it. Okay, so that's why what will happen is the until unless the 8 to 10 base pair it is amplified, it can be terminated before that. Okay, once this 8 to 10 base pair RNA will synthesize, then only the sigma factor will remove from this open conformation. Okay, and then RNA polymerase can continue the RNA synthesis from here. Okay, until that this sigma factor provide the strength also to not to terminate before that the RNA synthesis. Okay, so once this after synthesis of 8 to 10 base pair RNA, the sigma factor will release, and then the RNA polymerase will go for the elongation phase. Okay, where the free 3' OH will add the free nucleotide in that, and then it will continue the RNA synthesis. Okay, so here the first nucleotide on the RNA transcript is always purine triphosphate. Okay, that you also have to remember, like it is always purine triphosphate which will be during the RNA formation. Suppose this is the starting 5' in the nucleotide, so it should be always purine nucleotide. It can be either GTP or it can be either ATP, where all the 3 phosphate will be there. Okay, because it is the starting point. And after that what will happen is when new nucleotide will come, that time the phosphodiester bond will form between two nucleotide, right? So that time the gamma and beta phosphate will remove, and only alpha phosphate will be there, and it will be incorporated into the this mRNA. Okay, so the triple P, this is the triphosphate GPN OH structure remain on the RNA transcript until the RNA synthesis is complete. Okay, so here the first nucleotide will be having the triphosphate. Okay, so remaining all the nucleotide will be having only single phosphate. Okay, so and the 3 molecule form a transcription initiation complex. Okay, so this is called as the transcription initiation complex, where the RNA is little bit, means it is around 8 to 10 base pair the RNA is formed. The RNA polymerase is attached to it, and there is DNA is also there. So there is this complex is called as the initiation complex or tricomplex. Okay, ternary complex. So for the RNA synthesis there is no need of the primer, and the sigma subunit falls off from the RNA polymerase once the first 3 to 5 diester bond is formed. Okay, and the core enzyme moves along the DNA template to enter the elongation phase. So once this sigma subunit will remove, then this RNA polymerase got stable on that DNA template, and it will move for the elongation phase. Okay, so in elongation phase, what happens is the release of the sigma subunit causes the conformational change of the core enzyme. Okay, because now only the core enzyme will be remain in the RNA polymerase. Okay, because sigma factor is already removed, so this core enzyme will slide on the DNA template from 5 days to 3 days end. Okay, and because here you can see that there is a OH group. Okay, means suppose this is the adenosine. Okay, so it is having the OH group which can accommodate for phosphodiester bond formation. It require OH group, right? So it will act as a primer for the next nucleotide or coming nucleotide. Okay, so this 3 days end will provide the addition of further nucleotide. Okay, and free NTPs are added sequentially to the 3 days OH group of the NSAID RNA strand. Okay, so like that the mRNA will be elongated through the RNA polymerase from 5 days to 3 days direction. Okay, then the last step is the termination step. So in the termination step, like in case of the bacteria, there is two kind of termination. One is the row dependent, another one is the row independent. Okay, so row dependent termination is firstly the row factor will go and bind to the root site of the DNA in the RNA sequence. Okay, there is a site called root. Okay, so in root site this row factor will go and bind, and it will also slide until unless it will reach to the DNA RNA hybrid, and where the RNA polymerase is bind with those RNA DNA duplex. Okay, here, so this row is a kind of helicase. Okay, so what it will do is once it will reach here, like RNA DNA duplex, it got the signal like from here only the RNA should be synthesized. So it will, through helicase activity, it will break the RNA, and it will help to dissociate the RNA from the DNA duplex. Okay, and all the factor will be disassembled from this DNA, and it will be found as a complete mRNA. Okay, and in case of the row independent manner, what is happened is, the row independent manner, there is in this mRNA, there is hairpin loop will create because of the complementarity between two means same type of nucleotide. It will create complementarity, so that's why the RNA will be create one hairpin loop like structure. So when this structure will form, that time the RNA polymerase got signal like the RNA synthesis should be stop here. Okay, so this structure formation is act as a termination site. Okay, so when this will form, the RNA will stop, RNA polymerase will stop the synthesis of RNA, and it will dissociate from the RNA template, and secrete the like single strand of the mRNA. Okay, so this is the complete procedure of the transcription of bacterial transcription, we can say. Okay, so in the first step, in the initiation step, like the RNA polymerase is having the sigma factor which will recognize the promoter sequence. Okay, and in the promoter there is two sequences, one is -35 sequence and another one is the -10 sequences. Okay, and these sequences are the consensus sequence. Consensus sequence means if you are comparing two sequences, okay, two or three, suppose human, mouse, like that, if you are comparing the particular DNA sequences, okay, and of a particular site, so the concession sequence means each suppose in human if it is T, okay, so in the same position in mouse also it will be T only. okay, and other organism also it will be T. So like that there will be a complete motif, okay, which is called as the consonant sequence. Like here the T T G A C A, like that, okay, T A T A T T type of thing. Okay, so this is called as the consensus sequence. So this consensus sequence will be recognized by the sigma factor, and once this is recognized, then RNA polymerase go and sit on that particular site, and it will start the synthesis of RNA. And this particular complex is called as the ternary complex, because here DNA, um, um, yeah, DNA and RNA is little bit started for synthesis, and the RNA polymerase is also there. So it is called as the ternary complex or the transcription initiation complex. Okay, and the first nucleotide for RNA synthesis will be always purine triphosphate. It can be GTP or as well as the ATP. Okay, and there will be like first nucleotide will be having triphosphate, and remaining will be having only single phosphate. Okay, and it is having the 3 days OH group will which will occupy the next nucleotide. Okay, and like that the elongation will happen, and once the elongation is um, um, carry forwarded, one sequence will suppose here like the termination will occur, where the row factor bind with the root sequence, and once it will reach to the RNA polymerase, here this row factor will get signal like here I have to stop the RNA polymerase to further synthesize the RNA. So here after reaching here, it will do the helicase activity, and it will separate this RNA from this DNA duplex, as well as this RNA polymerase will also dissociate from this complex, and row factor will also dissociate from here, and again it will go into the cycle. And this in case of row independent thing, there will be hairpin structure will form in the mRNA, okay, due to complementarity. So that time the RNA polymerase will get signal like here I have to stop the RNA synthesis because the hairpin loop is produced. Okay, so this will help to dissociate from the DNA duplex, and then again this RNA polymerase will dissociate from there, and mRNA is also free from the DNA duplex. Okay, so these are the different inhibitor which inhibit the transcription process or the RNA polymerase process. So the first very common antibiotic is the rifampicin. Okay, so what it will do is it will bind to the beta subunit of the RNA polymerase and block the initiation of transcription. Okay, as we know that this beta subunit is helpful for the catalysis of RNA polymerase, so it will bind to the beta subunit, and it won't allow the RNA polymerase to start the transcription. Okay, and the next one is the actinomycin D. Okay, so this actinomycin D intercalate between the GC base pair, so that the elongation won't happen. Okay, suppose this is the G and C, like that sequence is there, so this actinomycin will bind here, so that the RNA polymerase can't move from G to C, right? So it will hamper the elongation process of the transcription. Okay, then you are having the cordycepin. Okay, cordycepin, it inhibit the elongation phase of the transcription. So here you can see that this is the analog of the adenosine. Okay, so once while synthesis of the RNA, when the in place of adenosine, if suppose this cordycepin is introduced, okay, in the mRNA template, so it is not having the 3 days OH group. Okay, here it is only having the H, it is not having the 3 days OH group. Like in case of the adenosine, it is having the OH group in the 3 days prime. Okay, so if it is introduced here, then there will be no chance to add more nucleotide into here. Okay, so it will also act as a inhibitor for the elongation phase of the transcription. Okay, so if it is introduced here, there is no free 3 days OH group, so that's why no more nucleotide will be added in the mRNA. Okay, so the transcription will be terminated there itself. Okay, So these are the different inhibitors that you have to remember, because in exam point of view it can be asked like rifampicin act on what subunit of the RNA polymerase, and which stage of the transcription is inhibited by cordycepin or actinomycin D, like that it can be asked, so that you have to remember. And this cordycepin is the analog of the adenosine, that also you have to remember. And is means the type of RNA in group 1 with their corresponding function in group 2: mRNA, rRNA, miRNA, tRNA. Here in the group 2 it is given serve as a adapter between the mRNA and amino acid during protein synthesis, regulate post transcriptional gene expression, codes for protein, form the core of the ribosome structure, and catalyze protein synthesis. What will be the correct option?
A.
A. Yes, the correct option will be option A, because mRNA is helpful for coding protein. The RNA is rRNA is the ribosomal RNA, so it form the core of ribosome structure and catalyze the protein synthesis. Then miRNA is the regulatory RNA, so it will regulate the post transcriptional gene expression, and tRNA is will charge the amino acid during the translation. Okay, so it serve as a adapter between mRNA and amino acid during the protein synthesis. Okay, so the correct option for this question will be option A. Okay, understood?